Okay, today we're going to do a blow-by-blow description of how to do three-factor crosses and also start some human mapping. I think people were getting the idea last time as we went through some of this, but I want to put it all together with an example and show you the step-by-step directions of how to do these problems. They're actually all the same, and they can all be approached in exactly the same way, but you need to know how to do each individual step to make it work out, and to do it efficiently so it doesn't take forever. So here's an example of a three-factor cross problem, and you can see that the progeny are listed in no particular order, and there's a whole bunch of different numbers associated with them. So the first thing we're going to do is basically follow the directions and follow my algorithmic approach to solving this particular problem. It looks overwhelming, but it's not. Okay, what you do is identify parentals, write as a heterozygote, then you identify the double crossover progeny, determine the gene order, calculate the distances, and draw the map. In some cases, you may be asked to calculate interference. Okay, so the first thing we do is identify parentals and write as a heterozygote. So the parentals are underlined here, vermilion hooked. We can identify those because they're the largest number. There's 301 of them. And its reciprocal is curved only, and that's 304 of those. So those are overwhelmingly larger in number than the other progeny types, so we know that that was the genotype of the original heterozygote before recombination had a chance to occur. Okay, so I'm writing these as a heterozygote in no particular order. I wrote vermilion hooked, VH, and then C+, because we assume that it's wild type for that particular gene. They like to confuse us that way. And its reciprocal is curved only, and so I wrote that as V plus H plus C, designating the mutant allele. Now, this is not the correct order necessarily. It could be, but this does tell you what the original arrangement of alleles was on the two chromosomes. Not in that order, but in terms of which homolog had which alleles on it. And when I write this, always write the gene and its own allele on top of each other. So I wrote V over V plus, H over H plus, C plus over C. So that's our starting point for what our parentals are and what the heterozygote looks like, irrespective of order. Okay, so step two is identify the double crossovers. Those are the fewest, and they're reciprocals of each other. Okay, so looking at our pile of numbers here, we have hooked, which is only 8, and we have curved vermilion, which is only 6. So those are going to be our double crossovers. They're much less frequent than any of the other progeny types. Okay, so I'm writing this, and this is not necessary, to be parallel to the heterozygote that I just wrote, where I put in the other alleles. That's not necessary. You could just write the mutant. You could just write hooked for one and vermilion curved for the other. But I like to write it in completely this way, just to be consistent with what I started with before. So that's not necessary. I think I'm maybe just being a little fussy here. Okay, so the crux of this now is to determine the gene order, because then we can tell which map distance that we calculate is which. So this is a systematic way of approaching it. There's also the kind of more intuitive by inspection way, but this one's good to know because it always works if you do it correctly. So I'm starting off with a vermilion hooked C+, and the V plus H plus C, which is what I wrote before when I identified the parentals. Now that could be the correct order, but it's only got a 1 in 3 chance. So now we write the other two possible orders just by sliding the alleles across the length of the chromosome. So before H was in the middle, now I'm just going to switch those and put V in the middle. I'm going to switch V and V plus both, and then put the H on the end to allow the V to be in the middle. We did not switch from one homolog to the other, though. We kept the wild type and mutant alleles on each homolog the same as what they were. We're just sliding them. So don't flip them from one homolog to the other. Slide them along the top or slide them along the bottom. And the third possibility is, of course, to have the curved in the middle. So I switch those two. I switch the curved and put that in the middle and put the other two on the ends. Okay, so those are the three possible orders. Now, top and bottom doesn't matter. I have them this way because that's how I wrote them at first. Those are equivalent to each other, so don't worry about that. Mirror image is also okay. You'll just end up with the maps in the different orientation. What really matters only is which alleles are in which homolog and which gene is in the middle. All right, so key here, we are going to decide which one of these orders gives us the double crossovers that we actually observed. Only one of them will work. The others won't work at all. So in this one, I have the V, H plus, C plus being one of the double crossover progeny, V plus, H, C being the other one. So the mutant phenotype here is going to be one is vermilion only, and the other is hooked and curved only. Now we look at our double crossovers, and we see that one of them is hooked only, and the other one is vermilion C. So that does not match the double crossovers that are given by this particular order. We don't have HC together, we have VC together. We have V and it's not by itself, so this one cannot work. Okay, we go to the second one, and we have H, V plus, C plus, and we have H plus, V, C. Okay, so we write that, and then we compare those to our actual double crossovers, and we see that this one does work actually. Now we're looking for phenotypes. The order in which these are written is not important. What we're looking is to see what mutant alleles are together and what wild type alleles are together. So this one works because it has H only, and it also has the VC together. So this is the correct order. The third possibility also doesn't work. It gives us VCH, V plus, C plus, H plus, right? And that, of course, does not match the double crossovers that we see, because this is all mutant and all wild type. So we can tell from looking at this that this center one is the correct order with the alleles written correctly on their homologs. So now we can look at what we can make a map out of this, because now we know which distance is which and what is where. Okay, so I've conveniently circled this here. So we've got, this is the correct order and the correct arrangement of alleles, and we can see that the double crossovers match. Okay, I put little arrows here to say by inspection, if you want to try doing that, that these alleles are together and these. So what you do is you look for one that's different. For example, in this example, we have the hooked only and the V plus C plus. Okay, so if you look at the actual alleles that are on that particular pair of homologs, you can see one matches, and the one that doesn't match is the V plus. Here we have the H, V, C plus, and we have H, V plus, C plus. The V plus is the one that's different, so we're putting the V in the middle. The other one, similarly, is H plus V, C, and this has the H plus C, and then the V plus is in the middle, so the V is the one that's different. So that's consistent with what we found before. The easiest approach to doing these actually is if you can do it by inspection this way to make a guess what you think is the correct order, and then check it to see if that actually gives you the correct double crossovers. Is probably the most efficient approach, although not guaranteed to work for you necessarily all the time, because it's easy to get tangled up in this sea of pluses and things. Okay, so now that we know what we have, we can draw the same heterozygote three ways. We're going to put the mutant alleles together with where they were, and then we're writing them in the correct order with the V vermilion in the middle. So now we can look at our progeny and see which numbers go with those map distances. So here we can make a single crossover between H and V, and the progeny of that will be H, V plus C, H plus V, C plus, and those, if we look at our progeny types, we can see that those are 71 plus 69. Now we have to count the double crossovers as well, because those also had a crossover in this region, and those are 8 and 6. So we calculate the map distance to be 71 plus 69 plus 8 plus 6, which is 154, and out of the total progeny of 1,000 gives us 15.4 map units. The next distance is the V to C distance, so we do that the same way. We write what the crossover products would be for that, and that's going to be H, V, C, H plus V plus C plus, and then we look at our progeny and see which ones match, and we see that's 121 and 119. And again, the double crossovers also have to count, because they had a crossover here too, so we add in our 8 plus our 6, get 255, gives us 25.5 map units. Now we're done. We just draw our map. We have H on one end, V in the middle, and C on the other end. And our first map distance, the H to V distance, is 15.4 centimorgans or map units. and the V to C distance is 25.5 centimorgans, or map distance. See, not so bad. Okay, so just follow the pattern. The homework example is due on Monday, sort of. Okay, so now we're going to move forward and start talking about human mapping, at least at some level. This is a very huge and complex field, and we're just going to hit some of the high points. Okay, one interesting thing about human mapping is that it seems like we don't need to do that anymore now that we have the whole genome sequence, which is certainly true. But there are a lot of diseases where it's not actually known what the affected gene is. So a lot of mapping is going to have to do with associating disease phenotypes with locations in the genome. And so there's a lot of mapping that goes on now relative to that whole problem of identifying human disease genes. Okay, so the mapping usually does not use phenotypes, and we don't do reciprocal crosses, and we don't do crossing times a homozygous recessive, because obviously we can't do controlled human crosses. So we're kind of stuck with what nature gives us. And moreover, we don't usually use actual genes and phenotypes for maps, except when we're discussing a disease locus. So the simple sequence DNA is generally used, including the short tandem repeat loci and a large number of SNPs, single nucleotide polymorphisms. So we're going to genotype a bunch of people, and we can use PCR microarrays or DNA sequencing to do that. We also have to realize that because these are DNA markers, nothing is recessive. We can basically see all the alleles if we have a PCR test or any other type test to identify which alleles are present, because both alleles will be visible to these DNA-based assays. So the idea is to locate the various SNPs and STRs relative to each other to make a map of the chromosome. Okay, so I like this as the earliest, part of the earliest human map available back in 1996, and it actually revealed some fairly startling features of the human genome. Here's our genome map, part of chromosome 1, and we can see that the map distances between the various STR loci are listed along the length of the chromosome, each individual map distance denoted between them. Okay, so one of the startling aspects of this was that females and males seem to have different lengths genetic map, which is to say that females have more recombination than males do. We normally see the sex average map, which for this chromosome is 272 centimorgans. The female equivalent is 363 centimorgans, and the male is 187 centimorgans. So females in general undergo more recombination per physical distance than do males. All right. So I already said this part. All right, so females have higher recombination than maps. I should point out, for sure, this should be obvious, but I'll point it out anyway, that the DNA sequence is the same for both sexes. So the physical map, which is to say how many actual base pairs separate two genes or two SNPs or two STRs, is the same regardless if you're male or female. It's just that that distance will experience more recombination if it's passed through the female germline versus if it's passed through the male germline. Okay, so it's good to take a look at this particular map to get a real sense of what's going on here. It's not a constant uniform difference from males to females. It's not like you can multiply by some constant and get the answer. That's not true at all. So it's good to compare the two maps side by side. So for example, D1S199 to D1S234 distance is 16.5 map units in females, but only 4 map units in males. Okay, there are some regions, however, which seem to have higher recombination in males versus females, and one example is D1S214 to D1S450 distance, which is 10.8 map units in males and only 1.8 map units in females. So you should look closely at these just to get an idea of the scale and extent of these kinds of differences between the males and females. Okay, well, this is not that surprising, but it does happen that our assumption, really, that the recombination rate would be directly proportional to the physical distance turns out not to be true, which is to say there's no constant which says, here's how many centimorgans there are per megabase in human. There are hot spots and cold spots, places where recombination occurs very frequently as hot spots, and cold spots where recombination frequency is really, very low. Okay, so there are very large differences from one region of the genome to itself, according to what the recombination rates are like. Okay, the centromere region is a famous example of a cold spot. This is megabases long, but crossing over is very rare. So this region is very small genetically, but is large physically, as measured by recombination versus DNA sequence. Okay, so here's one chromosome to represent this idea. The cytological map is written on the bottom or drawn on the bottom, where the characteristic banding pattern, Giemsa banding patterns of chromosome 12 is represented. The x-axis of the graph is the distance from one end of the chromosome to the other in base pairs. So this whole chromosome is more than 1.2 times 10 to the 8th base pairs long. So what's being graphed is the recombination rate at each megabase section of this chromosome. So each little spike, black spike, represents the recombination rate in one particular megabase region of that particular chromosome. Now, if the recombination rate were uniform across the chromosome, we would see something look like a lawn of grass where all the little black spikes were the same height. That is not the case here, obviously. For example, look at the centromere in the middle, that there's basically hardly any recombination at all within that region where the centromere is located. But even along the lengths of the arms is very variable in terms of how big the spike is. Like there's one region here in the middle of on the Q arm where the recombination rate is more than 60 centimorgans per megabase. But there are other regions of the chromosome where it's less than 5. So there's a huge variation in the different regions of the chromosome in terms of how much recombination they actually experience. So it's by no means a uniform recombination rate across the genome. And people are still working on trying to figure out exactly how that works and what's going on with recombination in those different kinds of regions. Okay, so this is what I just said. If recombination were equally likely at all locations, then the little black bars would be equal across the chromosome, and nothing could be further from the truth. All right, same as this. There is no uniform constant across the genome by which we can equate recombination frequency to DNA sequence. Okay, so moving on to how you track genes in the human genome, I just want to illustrate some of the principles and some of the vocabulary today before we get caught up in exact pedigrees and actually trying to track genes and diseases in pedigrees. Okay, so the idea is we're going to use sort of abbreviated nomenclature to refer to these loci, because the nomenclature like D1S something 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 allele 10 is really awkward to follow in a cross. It looks like just, you can't look at those kinds of things and be able to follow them very well. So abbreviating those loci as A, B, and C is a very easy way to take care of that. And of course, if you have STR loci, the alleles are going to be things like 10, 11, 12, 13. We're just going to use 1, 2, 3, 4, 5, 6 to refer to the number of repeats. Okay, these are easier to follow, they make the point, and they make it certainly a lot easier to look at when you're trying to figure out what's going on in a pedigree. Okay, so to illustrate what's going on here and why this is sometimes difficult and why we need to have often family information to figure things out, we consider a person who has a genotype for 5 STR loci, and we're going to call it A1, A2, B1, B2, C1, C2, D1, D2, E1, E2. So the person is heterozygous for alleles at those 5 loci. Now, if you try to follow this in a pedigree or trying to figure out recombination frequencies, you need to know something more than just a genotype, which is to say you need to know what the arrangement of alleles is in the particular individual so you can get the information about recombination or lack thereof. So just to illustrate this point, here's 3 different possible ways that those alleles could be arranged on a chromosome and give you the same genotype. Right, you've got A1, B1, no, A1, B2, C1, D1, E2 over A2, B1, C2, D2, E1, for example. Or else you could have all the 1s on one homolog, A through E1, and A2 through E2 on the other homolog, or a mixture. I mean, if you get a genotype from a PCR or a DNA sequence, it's not easy to tell which way, or sometimes impossible to tell, which way the arrangement actually is. So you have to have ways of understanding or identifying which are parentals and which are recombinants when you look in pedigrees to try to do any kind of gene tracking. Okay, so now we come to the idea of informative meioses and determining what's called the phase of the various alleles. So sometimes you're lucky and sometimes you're not. Some configurations can be figured out and others can't, just depending on what the actual information is in that particular family. So what this pedigree is about is trying to take the children in the third generation and figure out who's recombinant for the A and B loci versus who has a parental configuration. So in order to figure out who's recombinant and who's parental, you need to know what the parents are like. Now this is a particularly easy cross to deal with because mom is homozygous. She's A1, A1, B1, B1. So you certainly know that all of her children will get A1, B1. She has no other possibility of what to give. And then we have dad's configuration, and that's where determining the phase is important. Okay, so dad is A1, A2, B1, B2, but without other information, you wouldn't know whether the A1 went with the B1 or the A2 went with the B1, etc. Now, we can learn that by looking at his parents, fortunately. If we look at his dad, dad is homozygous A2, A2, B2, B2. So he must have gotten the A2, B2 from his dad, right? So we know that this parental configuration, A2, B2, came from dad, and that leaves the A1, B1, which he must have gotten from mom. So now we know what the parentals are. Knowing that, we can decide for each of the children which is recombinant and which is parental, because we know that his genotype is going to be A1, B1 over A2, B2, so that 1, 1 is one parental and the 2, 2 is the other parental. So we can look at the kids. This person has 2, 2, so he's non-recombinant. This one has 1, 1, he's non-recombinant. But this child is recombinant. He's got A1, B2, so there's been a rearrangement of the parental alleles in this child versus what dad had in the first place. Similarly, the fourth child is parental. He's got A2, B2. The next one's got A1, B1. And the next one's also recombinant because he has B1, A2. So the A and the B is switched during recombination in that father to give this recombinant child. And the last one is non-recombinant. He's got the A2, B2. So you couldn't unambiguously assign what these genotypes are relative to recombinant versus non-recombinant if you didn't have the appropriate information about dad's alleles and where those came from, and mom's alleles and what her genotype contribution to the kids is going to be. So it just depends on what the family looks like and what the pedigree itself, how much information is present. Okay, so this just goes through what I went through a minute ago. We know that the mother is homozygous, so her heterozygosity is not going to confuse anything. All of her kids are going to get A1, B1. The father is heterozygous for A1, A2, B1, B2. And if we didn't have information from his father, we wouldn't know which A went with which B. Fortunately, we can see from his father that he necessarily got the A2, B2 from his father, right? So that enables us to say that's the parental configuration for him. So his genotype is A1, B1 over A2, B2, with the A1, B1 from his mom, and the A2, B2 from his dad, right? It's also helpful, very helpful, that his wife is homozygous for A1 and B1, so if the kids get which alleles, we know what they're going to get from her, irrespective of any recombination event that happens in dad. Okay, so back to our original. For the dad, his configuration is A1, B1 over A2, B2. Now, I could have written this A2, B2 over A1, B1. It doesn't matter. But the fact that we know what the parental is means we can say whether there's been a recombination event or not to give the recombinant progeny in this particular cross. Okay, so the next thing we will be discussing is these kinds of situations where we're looking for information about linkage of a particular STR or SNP to a disease gene. And we're going to consider what types of meioses are informative versus uninformative to help us do these kinds of gene tracking. And we'll continue this on Monday. But it's good to know what the vocabulary is like before getting terribly confused by the actual pedigrees. Okay. See you Monday.